Hello, we are Group 10, and this is our presentation on the phosphoglucoisomerase enzyme. My name is Jade.
My name is Emmanuel.
And I'm Zhang.
My name is Vong Wing.
And I'm Haley. So today we will be discussing the name and classification of our enzyme, the structural and enzymatic features, its relationship to glycolysis, its mechanism and regulation, interaction with other pathways, clinical applications, deficiencies and disorders, and the effects on the body.
Phosphoglucoisomerase belongs to a process named glycolysis. Glycolysis is a multi-step process that has to convert glucose to pyruvate. And phosphoglucoisomerase is the second step of the process that has to convert glucose 6-phosphate to fructose 6-phosphate.
So for the function of phosphoglucoisomerase, this enzyme is crucial in not only the second step of glycolysis, but also the second step of the investment phase of glycolysis, where we invest 2 molecules of ATP to destabilize glucose and break it down into smaller units. So the function of the enzyme is to convert D-glucose 6-phosphate into D-fructose 6-phosphate. And we do this via an isomerase reaction where we open up the D-glucose 6-phosphate cyclic ring into the straight chain conformation, and we convert that hexophosphate from an aldose to the ketose conformation. And what that does, it exposes a terminal hydroxyl group, which can be phosphorylized to again destabilize that molecule, as shown below. So the importance of this enzyme, as we studied in the second unit of this course, it was used both in glycolysis and gluconeogenesis to interconvert from both the phosphoglucose 6 to the fructose 6-phosphate. It is a required step, again, in the investment phase of glycolysis to destabilize that glucose to break it down further. And it is also vital in yielding other precursor molecules that are essential for other pathways that we studied, as illustrated above. Some examples are the anaerobic and aerobic respiration, and TCA and electron transport chain cycle. So you can see that if we are lacking in this enzyme, essentially we inhibit various other biomolecule processes.
So the reaction completed by phosphoglucoisomerase is reversible, as evident by its very small delta G value. And this allows the reaction to proceed in either direction, and the enzyme is allowed to function in both glycolysis and gluconeogenesis. The enzyme is not necessarily regulated through an inhibitor or a kinase, but it is regulated through flux. This means that the enzyme maintains the equilibrium of glucose 6-phosphate and fructose 6-phosphate.
So next, we're going to talk about the phosphoglucoisomerase mechanism. As we know before, Emmanuel Haley talked about is enzyme that have reversible conversion of glucose 6-phosphate to fructose 6-phosphate, and it involves in an acid-base catalysis by the enzyme. The basic mechanism involves in the isomerization reaction of an aldose to a ketose, and this is performed by a ring opening and then followed by an isomerization of the open ring, and then the ring will be closing. We will take a look at more detailed steps in this slide. We can see step 1, the substrate will bind to the enzyme. Step 2, residue lysine or histidine will act as an enzymatic acid, catalyzing the opening of the ring. And step 3, we see conserved glutamate abstract the acidic proton from C2, forming a cis-endiol intermediate. And step 4, glutamate donates back the proton back to the C1 position. And final step 5, lysine or histidine will abstract back the proton from the sugar ring oxygen and restoring in a ring's closure to give the products as fructose 6-phosphate. We see here as a picture of the mechanism more detailed. We see ring opening, histidine will be donating the proton here, opening the ring structure, and we see glutamate will pull the proton from C2 to create the endiol intermediate, and also we see the ketone will create the double bond on C2, and ring closing reaction will be final step to create fructose 6-phosphate?
So, because the enzyme is not regulated, there are no natural inhibitors. However, in a structural study, multiple inhibitors were synthesized so that a crystal structure could be gained of the enzyme with its substrate bound. For this, the best inhibitor was 5-PAH, which is letter C in the diagram. And when this molecule was bound to the enzyme, they were able to get a crystal structure that showed the cis-ene-diol intermediate that Vaughn mentioned.
On this part, I want to talk to you about phosphoglucoisomerase and its structural features. Phosphoglucoisomerase is a 64 kilodalton dimer composed of two identical monomers. And phosphoglucoisomerase monomer are made of two domains, the large and the small domains. And two domains are each alpha, beta, alpha sandwich. On this picture, you can see that phosphoglucoisomerase has a large domain, the small domain, and the active site. The small domain contains a 5-strand beta sheet surrounded by alpha helix, and the large domain had a 6-strand beta sheet. As mentioned in the previous slide about the mechanisms, the active site is in the interface of the large domain and the small domain, and is the region with highest observed conservation containing several amino acid residues. On these pictures, you can see that there's several structural motifs to the protein. Some protein are made completely of alpha helix, some are made of beta sheet, and some are a combination of alpha and beta to make the sandwich structures, which is the structure of phosphoglucoisomerase.
Now, there are specific interactions of the substrates and products of the phosphoglucoisomerase enzyme with other pathways. Here in the figures, I've marked in yellow so you can see where glucose 6-phosphate can feed into the pentose phosphate pathway as an alternative route to form fructose 6-phosphate and reintegrate into the glycolytic pathway. Alternatively, glucose 6-phosphate can also redirect through glycogenesis pathway to promote the synthesis of glycogen. So what if phosphoglucoisomerase is not functioning properly? Well, this will prevent the conversion of an aldose sugar into a ketose sugar, which could hinder subsequent steps of the glycolytic pathway that are needed to break down glucose and produce pyruvate, NADH, and ATP as energy. Specifically, the lack of ATP and an enzymatic deficiency in phosphoglucoisomerase directly affects red blood cells and can lead to metabolic disorders within the body. Now, enzymatic disorders and deficiencies related to the phosphoglucoisomerase enzyme are typically hereditary. Disorders such as hemolytic anemia are inherited in an autosomal recessive pattern, so if a child inherits both mutated copies of the gene from each parent, they will likely experience symptoms related to erythroenzymopathy. This disease and enzyme connection can be made and explained through the function of red blood cells. The effects of a phosphoglucoisomerase deficiency in red blood cells destabilizes their membranes and induces their destruction in a process known as hemolysis. And hemolysis, in turn, will also decrease the ability of the red blood cells to transport oxygen to tissues and organs throughout the body. So the process of hemolysis significantly lowers the amount of red blood cells because more cells are being destroyed than created. The red blood cells burst and hemoglobin, the oxygen carrier, is released into the bloodstream instead of being directly transported into tissues. Since our tissues require oxygen to provide nutrients to all the cells in our body, the decrease can lead to many physiological changes and symptoms as listed here. In addition, hemolytic anemia exists in variable degrees, so for this reason, treatment options may vary based on the severity of the individual. Now, this is the end of our presentation. Thank you for listening.